To accelerate biological discovery, you need smarter tools. Tools that combine the precision of genome editing with the power of hybrid learning, digital twins, and sensors. This is the core of biointelligence. I am Marco Integhini, and this is Biointelligent, a podcast that decodes biotech scientific innovation within the Horizon Europe project BIOS and more. All right, so today I'm joined by Lorena Martínez España from VTT Technical Research Center from Finland. And within the BIOS project, especially Work Package 2 on digital twinning, VTT contributes to the processing modeling of the project, and Lorena's work focuses on the mechanistic bioprocess modeling. If I'm correct, you focus mostly on Pseudomonas putida cultivation under different conditions. And in this conversation, we will talk about modeling itself, digital twinning, biointelligence, and also what this experience has meant from the perspective of a PhD candidate. And well, thank you, Lorena. Thank you very much for joining me.
Thank you, Marco.
And to start, could you briefly introduce yourself and explain your role at VTT within the BIOS project and Work Package 2?
Well, my name is Lorena Martínez España. I am a PhD student, PhD researching in VTT. I'm in my third year of PhD. My background is in biotechnology, but during my last year of bachelor's, I discovered the world of substance biology, modeling, controlled, and I decided that that was my future. So I specialize in bioinformatics, and my research focuses on many things, but we can summarize at digital twinning, including different kinds of mechanistic models, OD models, genome scale models, also AI control, model predictive control. So everything related with model of bioprocess, kinetic modeling, control of bioprocesses. And in the framework of BIOS, I'm mainly in Work Package 2, developing the bioprocess model of the cultivation of Pseudomonas putida in different setups, also collaborating with other partners that develop other kinds of models to make a more, let's say, holistic kind of model.
And this is part of, well, it connects to the wider Work Package 2 in a whole, right? So it's not, okay. Where, well, different modeling are combined together.
I have collaborated with INRAE, combining machine learning and my process model. Also with the University of Wageningen, combining different bioprocess models for measuring different compounds and making control. There's this collaboration between work packages, because I think machine learning is a different one.
All right. So what is the main goal of your bioprocess model for Pseudomonas putida cultivation, and what problem is meant to solve in practice?
The bioprocess model of Pseudomonas is, as you have said, a mechanistic model that is built on ODEs, ordinary differential equations, and what we measure is how things evolve over time. So what we wanted to do is, let's say, make a drawing or what is happening in the reactor or in the page reader with equations that give us the opportunity to simplify the process, because in biology there's many factors you have to take into account, and it's very complex. What we are working on are very highly nonlinear processes, very highly complex processes. So with our equations we simplify those processes, and we have an image in our computers, and our goal is to predict what can happen in the bioreactor if we change the conditions we are studying, and we not only predict, but we also extract knowledge, because if we make different simulations with our models of different conditions, we can understand why the changes we are observing, where do they come from. So our main goal is prediction, obtaining knowledge, let's say expanding the borders of knowledge about a process, about a microorganism, about a compound, let's say, and also like using these models to optimize bioprocesses or control bioprocesses. That is the ultimate goal.
All right, so is the main value here better prediction, better control, or better experimental design in a way?
It's everything is in a pack of things, because we develop the model with the knowledge we have about the behavior of a microorganism, and once our model is accurate, is good enough, we can design experiments. So we don't have to make all the experiments, but we can optimize which experiments will give us more information. For example, let's say at which pH the Pseudomonas will grow better, how many glucose do we need, what is the quantity of CO2 that we are producing, the quantity of oxygen. So our goal with the model is not just having better predictions, but having better predictions, but also better experiment planification, better optimization of the processes, better control. So it's like a large field where the model is, let's say, the center of all of those fields.
Okay, so yeah, it's a combination of all of the above. And what are the key biological process variables your model tries to capture? You already mentioned something, and what are important for building a useful digital?
Yeah, as I mentioned before, a model is always a simplification of the reality. Sometimes we simplify because we don't have information enough. At this point, machine learning is really useful. Or also we can simplify because simply we can describe everything, because we will have huge models, because biology is complex. So for this bioprocess model, and based on the information we had from other partners and the literature information, because Pseudomonas putida is a quite relevant microorganism in industry, we wanted to study how glucose and pH and different setups influence the growth of Pseudomonas. For this task, we made experiments with different concentrations of glucose and at different pHs, and in two different setups that are small bioreactors and plate readers, and see how Pseudomonas was growing, and also how the metabolism of Pseudomonas was behaving, because using HPLC we measured the internal metabolites, so we can see how these internal metabolites change over time, so we can see the effect of pH, glucose, and different media, not only affecting the growth but also affecting the main metabolic pathways in Pseudomonas, and that is what we wanted to do and the knowledge we wanted to have. What happened when we have different pH, what happens if we control the pH, that is something that we also include in the models. We can control the pH computationally as we have in our reactors, or we can let the pH change freely as we have in plate readers.
All right, and how is this process going? So what are the preliminary results you can already share eventually? And something I'm curious about, is there one of these variables which was most difficult to predict? So which was the hardest part to model in this context?
Well, for me personally, it was a challenge, because usually when we model some organism, we have people that have modeled some other things about this organism before, so we can study a bit what other people did before, and then you try to, let's say, think out of the box. But in this case, there's a lot of literature about Pseudomonas putida, about the metabolism of Pseudomonas putida, because it has a quite interesting. Metabolism is really useful for many biotechnological industries, but there was not a bioprocess model of Pseudomonas before. So from my experience as a researcher, modeling a carbon source is not really complicated because many mechanisms are already known. But in the case of pH, we are using media that had a lot of chemical compounds, and the simulation of the changes of pH, how each salt of the media affects the pH, also how the gases affect the pH, the growth of Pseudomonas is affecting the pH, was quite challenging. I am really lucky that in my team I had the help of Peter Blomberg, that has large expertise in this field, and I am very thankful I could learn from him. So that was, I think, the main challenge of this model, trying to develop a pH calculation that was accurate with the results. And now we're in the last part of the modeling part, that is the parameter estimation, that maybe is the most difficult part, but maybe difficult mathematically. But for the results we have now, they are very promising because we are able to predict quite accurately the growth of Pseudomonas in different conditions. So it seems the model is working, so the model could be used in the industry for the cultivation of Pseudomonas. And of course, there's a lot of work to do in other scales, and there's many other things we can take into account, but I think it's a good start.
Well, it sounds like a good bio success story
Yeah.
so far.
Yeah.
And like having a broader point of view, how does this mechanistic model, the one you worked with mostly, connect to the broader bias vision on digital twin, and especially when combined with other machine learning and what's the name again?
Physical informed neural networks. I must say that BIOS project is super diverse. Like, there's people doing many stuff, and we are very lucky because we had people working on different kinds of modeling. As we mentioned, a digital twin is a drawing of reality. We are trying to simulate what is happening in reality, and my model is just a piece of that drawing. Because I have the model of the cultivation, but then there's models, genome scales models, of the metabolism of Pseudomonas, so we can look inside the cell what is happening. And nowadays, it's clear artificial intelligence is a tool that is extremely useful, there's no doubts about that. And physical informed neural networks, they were created not for biology originally, as machine learning was also created for physics, but it's a really nice tool that can help us to predict the parameters of our model, because that is maybe the bottleneck of my field, the parameter estimation, that is finding the value of the parameters of the equations that makes that my simulation and the experimental data, they match. That is made through optimization methods of non-convex problems that are really difficult to solve, really computationally heavy, and specifically pins. They have the power to help this process to be faster and to be more accurate, with maybe the disadvantage of the need of big amount of data. But as I mentioned before, BIOS is a super diverse project, so we had large amount of data, not only in BTT but in different partners. Also the University of Wageningen produced a lot of data that is used to train the pins. Last year I had the opportunity to make my research stay in Paris with the partners of INRAE working in these pins, and it was really nice to learn more about this topic, because machine learning is something really broad. So I think we are doing a good job with INRAE, and I think it could be, I hope it is another successful case in BIOS, the combination of machine learning and bioprocess model to predict parameters and to push this prediction power we have.
All right. Thank you for this more integrated view. So like all these different approaches that are complementary at the end, did you consider them complementary from the start, or is it something that you realized while doing it? Like you were maybe just focusing on your part at the beginning and then you realized how well connected they are, or you already had a larger view from the beginning?
No, really from the beginning, I realized we needed to combine different models. Thanks to my studies, I studied different kinds of models, how to work with different kinds of models. Also in my research, I work in other projects that are not BIOS, where I use, for example, genome scale models, these metabolic models. So from the beginning, I knew that at least these metabolic models should be connected with bioprocess models. Let's say the bioprocess models we look at outside, what is happening in my bioreactor, and with metabolic models we can look at inside. So we have a more complete view, but it's true that in the recent years we see how artificial intelligence, we have new advances every day, we have new papers every day. So I really didn't know about physical informed neural networks until I went to Paris and I had the opportunity to learn about them. And at that moment, I really thought that they are needed. We also need to improve the methods of machine learning, because the improve of artificial intelligence will improve other fields, because the prediction power they have is huge. And if we include that prediction power with mechanistic models, we make our knowledge bigger. That is the main advantage of combining the knowledge acquisition that we have with mechanistic models with the prediction power of black box models like machine learning. So I think it's nowadays it's really clear, like all the modelers, we should solve between us to combine them. And I think BIOS has been really good at doing that.
Nice. Well, that's a very nice point of view, also considering the AI integration with physical informed knowledge and of course, we want to make some knowledge out of the black box, and this is a good approach, as you mentioned. Connected to this, but well, not so connected. This is a slightly change of topic, but still in the same area. What does biointelligence actually mean in this context, and from your point of view?
If I am honest, like I knew the concept biointelligence with BIOS. And during all this time that I've been in the project, I think is like a collage of things, because it goes beyond prediction. I think nowadays the world is very fast and the science is very fast. I mentioned it, we have new papers every day, new methods, new approaches. It's very fast, even that fast that we don't have the real time to realize the importance of those methods. And sometimes with this situation, we forget that we are here to have new knowledge, to expand the borders of knowledge of science. So for me, biointelligence, it's a framework that integrates predictive modeling, every kind of modeling, decision support for scientists, mechanistic understanding, try to, let's say, put new glasses and see what is happening inside the cells, like understanding why the organisms are behaving the way they behave. And with all of this, in the case of industrial biotechnology, we can make bioprocesses or processes more controllable, so we can reach our goals in production. That it is, I think, the ultimate goal of this field, that making life better through producing new things, discovering new things, but also more interpretable. So we have more knowledge about what is happening. So on the one hand, we anticipate the system behavior because we can predict. We know what will happen in our bioreactors because before we have integrated different kinds of modeling. People from different disciplines joined, like in BIOS, to anticipate this behavior, and then over that prediction we obtain more knowledge, knowledge that is very valuable for the scientific community. Also with all this literature of fundamental science that sometimes is underestimated, we give that knowledge, like, okay, now we know a new process that is happening inside the cell. We discover new metabolites that pseudomonas can produce. So for me, it's the mix of prediction, knowledge, control, and the success of bioprocesses and the success of production.
Yeah, like, it's always nice to hear that we are in this fast advancing world, and science, technology are being produced. New technologies are being produced so fast, as you mentioned, but it's always important to think about fundamentals first. So nice that you mentioned it. Now, I would also like to move a bit to your own personal perspective as a researcher. So I would like to know if I would like to know, as a PhD student's point of view, how was working with this modeling challenge. Well, you already mentioned that you got a lot of inspiration by the people that mentored you, but I would also like to know how it is working with this international consortium like BIOS, and how do you think this is contributing to your scientific career so far?
From my point of view, now if I look at myself almost 3 years ago, and I see that person, as I mentioned, like I feel extremely lucky of being in BTT, in my team, because I've learned a lot. Like now I look at my equations and I think, wow, you didn't know that 3 years ago. Like you didn't know many dynamics. Sorry. You didn't know many processes. You didn't know many math. And now you know them, and now you can do it. And it's thanks to all the people that mentor me and help me and take time to give me the opportunity of learn. So it's a very nice feeling as a PhD student, feel that you are becoming a scientist. And also BIOS is my first European project as a researcher. And as I mentioned, I think the consortium we have, it's incredibly nice. It's amazing. Many people from many backgrounds, computational, experimental, working together in many things that may seem different, but they are really connected. Biosensors are connected with modeling and with neural networks and with production and with development of methods. Everything is connected, and when you see that over the years, you see the progress, you see the new discoveries, you read the papers, you see the conferences where all the consortia attended. It's amazing, and also it gave me the opportunity to learn how to network with people. I think it's something really important for scientists, having this network of scientists, and having the opportunity of doing research stays in Paris and also in University of Wageningen during spring this year. It really helped me to grow as a scientist, not only because of the science we created, but also to see how other people work in their labs, how are the dynamics in different universities, working with not only, like, let's say, the PIs or the team leaders, but working with other students, try to find solutions together, brainstorming sessions. It's something that you don't realize, but it really has an effect on your way of thinking or your way of making research. And I think the experience has been amazing, because now I see that I am becoming a scientist little by little, and the partners of BIOS contribute to this, and the knowledge generated in BIOS contributed to this, and I am sure it will contribute to society to solve real problems. That is the goal of science, like solve these problems and anticipate to the future problems.
That's amazing to hear, because most of the time in the scientific field we focus on the outcome of the research itself, but it's always nice to consider that this project also form people. They could help grow scientists, and that in turn will develop and will research for more things, more. They will develop more projects and
Exactly.
so on. So thank you for sharing this
Yeah.
this point of view. That was very nice. I'm happy that you mentioned. Yeah, okay. So connected also to your developing career as a scientist, and like the last question to conclude our discussion would be, looking ahead, what are the next step for the model specifically, but also in general, the next step for your research within BIOS, well, eventually in future, if you have something to share.
Well, now we are in the last step of the model development, that is the parameter estimation. So we are running those processes that, as I said, they are computationally very heavy, and more with this model that is more than 20 equations, like really heavy model. But the parameters we obtain so far, they are very nice. The model is predicting nicely. It's identifiable. We are not having problems with the parameters. So we are in the step of just finalizing the simulations, writing this paper that will describe will present the model and all the experiments we have made. We are still collaborating with INRAE, with these neural networks, also collaborating with the University of Wageningen to the merge of different models and control. So yeah, it's the end of the project, but I think it's the most busy months for me, because many things are going on, and now that I have all the knowledge is when all the ideas come to my mind, like all the things I want to do, because there's many things to know. So yeah, hopefully we will have all these papers out. I am presenting our research in the European Conference of Computational Biology in Switzerland this September. That is
Nice to hear.
also, it's also really cool, like to present your work to that conference with like important researchers and people from different fields, it's always exciting, and also see what is happening in the computational biology world. Also other topics that are not for my research. But yeah, with these papers and my other papers of other projects, hopefully I will become a doctor next year. And yeah, like from BIOS, I really have some partners that I will really want to collaborate with in the future, like in European proposals or in different calls. It's something I am also very interested in, and my supervisor, Dorothy Barth, is helping me with this process of learning about calls, different projects, how to write proposals. So I think that is the next step after becoming a doctor, that is applying for funding, and I am really excited because now I know a lot of people and I know different labs that are doing really, really impressive science and that I would like to collaborate with. So yeah, BIOS made nice science, but I think it's also making nice scientists, and it will make more projects will come out of this one, so it's really nice. So hopefully, if everything goes well, that is the plan.
Nice. That's an amazing overview. I wouldn't know how to say it in better words. So we have good science, good scientists eventually, and follow up. So yeah, that's an amazing summary you made. Yeah, well, I guess that's lead us to the end. With this, I would like to thank you, Lorena. Thank you very much. This was very clear and insightful look on both modeling and your personal perspective. And yeah, you shared a very nice point of view with us. And yeah, thank you for joining me, and see you soon.
Thank you, Marco. Thank you very much.